Hello, everybody. You have clicked in a series of teaching slide presentations in Zoom conferences explaining clinical drug development in oncology. This time, we will talk about modified toxicity interval design, mTPI. The video goes over the context, explains how to handle an mTPI table, how to create it from the viewpoint of the clinical scientist or medical director, and gives information how to find more information if you want to go deeper in the statistics. My name is Johannes Wolf. This is recorded in May 2024. mTPI is a further development of the 3+3 design using Bayesian mathematics. It is easier to understand it when you understand 3+3 design. There was a 3+3 design video earlier in the series. If you've seen that, you don't need to see this and the next slide, flip over, go to 3 slides from now. Also, if you know 3+3 design, just scroll forward. For the others, hematology oncology is the medical field that treats cancer. Drug development means we are making new drugs, bringing them to patients to cure cancer or at least make it better. This is typically done in 3 phases. Phase 1 studies are those that find the correct dose. This is typically the maximal tolerated dose or a derivative of that, the recommended phase 2 dose. And the classical design is the so-called 3+3 design. Phase 2 studies look at preliminary efficacy. The typical endpoint is response. This means how many tumors shrank in response to giving the drug. And phase 3 designs are very large studies where large numbers of patients receive the new drug versus large numbers of patients receiving standard of care. And then the endpoint is overall survival, determining which of the two is better. What you're looking at here is a typical study design of a phase 1 study with 3+3 design. The doses that the patients received were increased from patient group to patient group. We call them cohorts from 25 milligrams to 350 milligrams. These are the data, and this has all been done in 3+3 design. 3+3 design enrolls 3 patients in a dose level. Then toxicity is observed. Now, toxicity is classified in a very simple parameter, dose-limiting toxicity. It is simplified to a yes or no. And then patients who have received the drug and experienced dose-limiting toxicity are counted. This may be 0, 1, 2, or 3 patients that experience dose-limiting toxicity among the 3 that received the drug. If it was none, then the 3+3 design thinks that dose is too low and it should go higher to the next higher dose level. If it was 1, then the design thinks this might be the right dose. Let's add 3 more patients to the same dose level. If it's 2 or 3, then the design says those are too many, the dose is too high, and then the lower dose can be tested if it wasn't tested yet. This keeps going until the next higher level is determined as being too high, and the next lower level below it has 6 patients enrolled, and among those 6 patients, there is 1 or no dose-limiting toxicity observed. That will then be called the maximal tolerated dose, and the study was a success and is completed. The study design is relatively rigid and has had many successes, but also many critiques. Further study designs have been developed. By now, there are many. This is a typical mTPI table as it actually occurred in a real protocol. You see the rows tell you how many patients were enrolled in a dose level, from how many patients there are data of potentially having or not having DLTs. Assuming there are 3 patients and you have data of 3 patients to evaluate, if there was no DLT among 3 patients, then the table tells you to escalate. If there was 1 DLT among 3 patients, then the order of the table is to stay on the current dose and enroll more patients. If there are 2, then the makers of this table think that dose was too high and the dose should be de-escalated. Now, mTPI allows you to go down and up again, so you can come back to an old dose level. That is different from 3+3 design and therefore requires further rules, a further category, and the category is the category of elimination. If there were too many dose-limiting toxicities in a cohort, then that dose will not be assessed any further. The dose will be eliminated. That is the DU. The dose is considered of unacceptable toxicity. Now with S, stay, we have a little problem in mTPI that is different from 3+3 design, because we need to know how many more patients should we enroll before we do the next dose level assessment. For this, within this table, you go diagonal until you reach a DU, the possibility of elimination. That is the number of extra patients you enroll. So if there were 3, 1 dose-limiting toxicity observed, then you enroll another 3 to come to 6. That is precisely the same thing as what 3+3 would have told you. So in a way, they are similar, except that these numbers of 4, 5, or 7 to 10 patients in a dose level wouldn't occur in a 3+3 design. But they're not exactly the same. Actually, in the real core of what they do, they're quite different. If you look at the box here, the red box, you see that 6 patients enrolled, 2 experienced DLTs. The table tells you stay. Now, that is different from 3+3 design. In 3+3 design, you would not stay on that dose level. That dose level would be considered too high. So this is, in a way, more aggressive. And it is even more aggressive if you look at the next one. Even if 3 patients experience dose-limiting toxicities among 6, the table would still tell you stay and enroll 2 more. See diagonal, you come here to DU. In 3+3 design, that is way too high. And that's what I thought when I saw this table for the first time, and I immediately asked the question until I looked at the definition of the dose-limiting toxicities in this very trial. And then it became clear. For somebody who is used to write and read and run 3+3 design studies, you also have a feeling and an impression, a gut feeling of how toxic does something need to be to actually make it a dose-limiting toxicity. And those are severe toxicities that we 3+3 designers call a dose-limiting toxicity. But if you read the definition of this study, you see that lower levels of toxicity were considered dose-limiting toxicity in this protocol. For instance, 3 days of grade 2 vomiting is a dose-limiting toxicity in this protocol, and it is not necessarily a dose-limiting toxicity in a classical chemotherapy protocol. Among chemotherapy treating physicians with 3 days of vomiting, you would think that is not good, and you would change something, but you wouldn't necessarily drop the dose. You would increase the antiemetics, you would do something to handle the toxicity and keep going with the same dose, hoping that the dose will help against the cancer. Here, it is considered a dose-limiting toxicity, but then the table allows you to carry on. So with that definition of DLT, this table makes sense. If with a classical definition of DLTs as it is for 3+3 design, this would be way too toxic. You see here one of the problems of mTPI. It has more choices. It is more precise. It performs better, as the statisticians will tell you, but with the choices come potentials for errors. Now, these designs don't stand completely alone. These designs don't stand completely alone. There are safety nets built into these protocols, and a safety net that you don't see in this table here is the Safety Review Committee. That is typically made out of experienced clinicians that have a completely different way of looking at that. They follow their own clinical instincts and don't care that much if something is called a DLT or not. They look at the toxicity and say, this is too much, or no, it's not a problem. Let's keep going. Those people would have stopped this if there were 3 dose-limiting toxicities as defined in classical 3+3 designs. And then they would have saved patients from getting too high doses in the future. But it is necessary to know that the safety review committee has to be there and has to be in power to override the statistical rules if need be. Okay, having talked about how to use these tables, let's now talk how to make them. The table is probably all you need to know to run a protocol with mTPI design. But if you want to design a protocol like that yourself and write the protocol, then it might be useful to dig a little deeper. What you need to know is that this was the paper that it was all based upon, and those are the authors of that paper. The first author is by now a professor at the University of Chicago. Those 2, by my knowledge, are still in Houston and maybe has moved to the industry. The paper has formulas like that, and if you can read them, then go ahead and read the paper. You may not need this video. I did not completely understand it, and I called my old friend Nebby, with whom I published before, and it was a delightful conversation, and he really helped me forward. So let me explain what I understand here. Understanding mathematical concepts often starts with understanding what assumptions were made. We are trying to find the right dose. The first assumption here is that there is such a thing as the right dose. And that is not always true. If you read different labels from anti-cancer drugs, you will find that within the same label, the dose may differ from indication to indication. Within the same indication, we often have protocols where the dose differs at different parts of the treatment protocol. For instance, there may be a ramp-up phase in the beginning, there may be a high induction dose, there may be a lower maintenance dose. Within the same indication, in the same cancer, the doses for different patients most certainly differ. Large people typically need higher doses than small people. Within the same patient, the dose may differ even within the same day. Think about insulin. The dose differs by the hours, and it is an art to find the right dose. Don't believe that a study protocol with a handful of cancer patients can find the right dose that is the right for everybody at all times. That is the work of the clinicians. As a drug developer, you should trust the clinicians that come after you. Don't try to make a point landing for a dose, because there is no point to land at. You only waste time and hold them off from doing their work. The second assumption is the higher the dose, the more toxicity. Now, that is often true, but not completely. It builds on the old sentence of Paracelsus. When he was asked, is this poison? He answered, only the dose makes it poison. Yes, typically the higher the dose, the more toxic will it be. But think about receptor-mediated processes. Once all receptors are occupied, the effect has been maximized, and adding more drug to it will not change the effect. That's true for both efficacy and for toxicity. Now, what the authors of mTPI design have done was to change the old classification of the infinite number of doses from a categories of 2, a binary, to a 3-category system. In the old system, we knew there are doses that are too high, they are toxic, and we tried to land as close as possible to that dose, believing that when you give more, you will also have more effect. Thus, give as much as you can. That paradigm is also changing. It comes out of old chemotherapy thinking, and modern drugs will change it, but it hasn't changed yet. mTPI now doesn't go for 2 categories, it goes for 3. Too low, acceptable, and the too high category hasn't changed. That is done by defining an equivalence interval, EI, as target toxicity minus epsilon to target toxicity plus epsilon. Everything below that is too low, everything above that is too high. Once that is defined, they calculate something that they call the unit probability mass, which is a ratio of areas under the curve before and after getting new data. That's where it gets complicated in the mathematics, but based upon those UPMs, then the decision is made for each situation to either escalate the dose, stay on the same dose, de-escalate the dose, or eliminate the dose level. There is more mathematics than the assumptions and the basic principle. If you like going deeper and you like watching videos, I have 2 recommendations. One was recorded in 2015 and is from my friend Nebi Beckerli. He teaches there a class of graduate students, and it has a lot of this human components of how was this developed, and you hear them ask questions and him answering. I love hearing the talk. The second one is more technical, more on the mathematical side, and is recorded quite a bit later. However, I'm going to say YouTube videos are not enough to actually understand this method. You're going to have to read the paper, and I'm going to go further and say even reading the paper is not enough to understand what the mathematics. You have to read the code. The paper has a free link to a code where you can download it, and where the code is in Excel and in R. Unfortunately, that link doesn't work anymore. The paper is from 2010. When I realized that, I called Nebi and he just sent me the code. It doesn't work for Excel because it uses macros that Microsoft has since abandoned and is not supporting anymore. The R code still works, but you have to have R on your computer. But I am actually not recommending any of my colleagues, the MD colleagues, the clinical drug developer, neither medical directors nor clinical scientists, to do that on their own. My recommendation is, if you don't have a fully educated statistician on your side, go with 3+3 design. If you want to do mTPI, have a fully educated biostatistician at your side, and then work this through. When you have a statistician at your side and you grew up through the medical line, you have a little problem because those 2 disciplines typically have a hard time to talk to each other. But that's what you need to do. So let me show you how I view this in the context of changing 3+3 to mTPI. 3+3 was this. We had in our art of drug development 4 regulators that we could change. The DLT period, if you make that longer, the study gets longer, you catch more adverse events, it gets more precise. DLT definition determines how early a dose-limiting toxicity is defined as opposed to other adverse events that are not considered dose-limiting. And if you make that tighter, then less DLTs will be determined. The maximum dose determines when the study stops. If you don't see any adverse event in the starting dose, determines where the study starts. And if you put this too high, then you will end up with a lot of toxicity in the beginning, and that kills the entire drug development. mTPI has the same regulators to use in our art, but now there are more regulators which are done in the art of the statistician. But the statisticians will ask you questions. And it is a good idea to be prepared. So one possibility in Bayesian is always, are there prior data? Now, the default for a first-in-human drug development study is to use non-informative beta, which kind of means you don't need to answer that question. But this one you have to answer. The statistician will ask you, what is your target toxicity? And please don't get offended. That doesn't mean that they ask you to have toxicity. That doesn't mean they want you to ignore your first lecture in medicine, where it says, first of all, do no harm. Our target toxicity, obviously, as physicians, is no toxicity, zero. That's not quite true in the context of chemotherapy, and that is the context where this needs to be seen. The target toxicity rate, the default would be 30%, and it would mean that you accept 3 in 10 patients to have dose-limiting toxicity by the definition of from before. The next one is even harder to intuitively answer, the equivalence interval. Now, I can only say use the default of 0.05, see what comes out of it, and then try to change it. The minimum cohort size, the default is 3, but it could be smaller or larger. The maximum cohort size is typically something between 6 and 12. If you make the minimum cohort size 3 and the maximum cohort size 6, then you're almost back to a 3+3 design. This is another way of showing how your impact to the questions of the statistician will impact the table. So this is, again, the table I showed in the beginning. And you see the minimum patient number obviously simply cuts off the table that you saw in the paper on the left side. The maximum patient number cuts it off on the right side. Those are easy to understand. Now, the target toxicity is almost easy to understand. You see, this has a target toxicity of 30%. And if you see 10 patients enrolled, 3 patients DLTs, that is in the area of S. S means stay. This is sort of the acceptable area. So that is also understandable. If you were to pick a higher number, then that area with S stay acceptable will move down. If you pick a lower number, it will go up. The epsilon, the equivalence interval, is the hardest thing to understand. The only thing I can say is it influences the width of this area and also these other numbers. And you can also say, while I said the standard is 0.05, in this protocol, the authors actually took 0.03. It's not intuitively simple to say what happens when you change that number. My recommendation is to test it out. Give a number, look what it looks like, and then see if it makes clinical sense. In addition to this, once it is perfect and the protocol goes to the FDA and the FDA says, yes, fine, go ahead and you enroll patients, you have to continue watching, because medicine, human patients are more messy than binary code can ever be. And you cannot get it always right for all patients. You have to watch it, you have to see the effect, and you have to potentially overwrite the beautiful statistical method because all of a sudden something new came up that nobody could have seen before. Congratulations, we're coming towards the end of the video. There are no more information to be absorbed and memorized. This is just a comparison as a way of repetition between mTPI, the new design, and 3+3 design, the classical old design. Both of them have strength and weakness, and then altogether, I actually don't think they are all too different. So let's go over the strength of mTPI. It clearly is a wonderful, flexible design that gives you a lot of places where you can change things and get the protocol right. I also think, at least compared to many other designs that came later and are even more sophisticated, mTPI has this table, and the table is easy to follow. I think that is an enormous strength, and I am thankful to the authors that they came up to this point. There are weaknesses with both of those designs. First of all, there is the assumption that there is one dose for everybody, and that is wrong. We can only have somewhat of a vague understanding of an average dose. The individual dose still has to be adapted to the individual patient. Similarly, there is an oversimplification of adverse events, side effects, toxicity of drugs as a simple binary signal as DLT, yes or no. Reality is far more complicated. Probably the biggest problem is that the method does not look at efficacy. It looks at toxicity only, and that is a reminiscence of the past where we developed chemotherapy. I hope that we will move on from this in the future to have also elements of efficacy when we look at the dose. And then the study design remains a bit complex and hard to understand. If you're not a statistician, I would count that also as a weakness. If you compare that to the old 3+3 design, it has pretty much the same things. There is a strength in 3+3 design that it is far more widely understood that the mTPI, if you are in an elevator and you meet the CEO and you have to explain why you go to the next dose up or down, that person will understand when you're talking in 3+3 design, but the elevator ride will not be enough to explain why that is the case in mTPI. 3+3 design is also very easy to follow. The big disadvantage of 3+3 design is that the protocol design is rigid. These are the disclaimers and conflict of interest. There is actually no conflict of interest. The video is brought to you by Oncology Consulting Wolf LLC. This is my own company. Nobody influences what we're saying. Unfortunately, we also don't have any sponsors. The science moves on. This video was made in May 2024. Things change, opinions may change as well. What you see here is the reflection of the opinions and knowledge at that time. I have a medical license, but obviously watching this video doesn't constitute a medical physician-patient relationship. If you have not been a patient before, then watching these videos does not make you a patient. And if you have been a patient before, our relationship as physician-patient is lifelong, and watching the videos also doesn't change this. I thank you very much. You have a wonderful day. Please come back to the next video.